Yep, we use that for stomach parasites, and this is good for wound healing, and this is good for fever. It was literally like walking down the forest path was walking down a pharmacy for them.
Welcome to the Knowledge Archives podcast. We're a group of students on a mission to learn from as many different disciplines of knowledge as possible. Today, I'm glad to be joined by Dr. Josh Kellogg, an assistant professor at the Department of Biomolecular Sciences at Pennsylvania State University in the US. He has what I think is the coolest job in the world, discovering new drugs from nature while collaborating with indigenous cultures from all around the world. Today, he explains what he does, what he's found, and what he's thinking about next. I really appreciate you taking the time to join me today because, as I said, this is just a very personally fascinating area for me. But first, to just share a little bit about, who you are and how you got interested in this field, it would be great to hear in an introduction.
Well, thank you for having me. And my name is Josh Kellogg. I'm an assistant professor at Pennsylvania State University out here in State College, Pennsylvania. And our lab focuses on medicinal plants and natural products. So we're looking for new medicines from nature, broadly defined. Most of our work is on plants, but we also do some work on fungi and bacteria and other natural resources. Yes, I'm a chemist by training. I loved chemistry growing up. I was the one in high school and college that really thought it was fascinating when everyone else hated it. But when I was thinking about what to do post graduation, I wanted something that combined my love of nature. I'm a big hiker, camper, biker, kayaker, etc. And I had taken one class as an undergrad that was an ethnobotany class. So ethnobotany is the study of how cultures around the globe use plants in their daily lives. So that it could be plants as food, as medicine, as construction, ceremonial. So there's a lot of facets to it. But my interest was really on the nutrition and the medicinal side of things. And so this class I took was all about how these plants that cultures had used across the globe, from South America to Africa to North America, Asia, how they actually had really interesting chemistry. A lot of them could be or had already been transformed into medicines. And this just clicked with me. It floored me that this sort of research was out there, because honestly, it hadn't really occurred to me. And so as I was thinking about going to grad school and kind of, continuing on in research and science, this really connected with me and lit the fire as to what I wanted to kind of spend my time researching.
Yeah, I just think that's the greatest career ever. You get to help people, you get to make cool scientific discoveries with drugs, you get to spend time in nature, you get to work with lots of cultures. I bet you probably get to travel, and I'm sure, there are more complexities than just those highlights. So could you guide us through the process of going from a plant out there in the fields to, having a new drug?
Sure, you were correct. It is a long process. I think one of the longest parts of that is finding a plant or fungus or some natural resource that has really promising activity. So really promising potential as a medicine. And there are different ways to find those plants. There's some groups you can go and just collect everything you see just randomly. You're hiking along the path and you pick every different kind of plant you could think of and bring them back and try them all. And that's one way to do it. That's very comprehensive, but that is a long process because you don't have any rationale of why you're picking plants and such. Some people use the relationships between plants, the families of plants, to guide their searches. We know that certain families are very potent, so like the Solanaceae family, where tobacco is from and things like that, like they have a lot of potent chemistry. So if we find a plant that is in the same family, maybe that gives us an idea that there's something worthwhile studying. A third way is to use traditional knowledge. So working with communities to understand which plants they used for different ailments and using that as a rationale to guide your search. And that has been shown in a few studies to have a better positive response rate. So you get better chance of finding potentially medicinal or bioactive plants if you make use of this ethnobotanical, this traditional ecological knowledge. Nowadays, there's also more modern techniques. Like we can collect plants, and rather than testing them, you can do genomics. You can map the genomes and look for particular genes that code for production of these molecules that might be bioactive. So there's definitely more, you could say, modern technologies or techniques that we have that scientists and other groups have developed that help in the screening process. But that also can be very labor intensive and sometimes quite expensive. So nothing is without its detractors. But once you have a plant that you've identified that you think is going to work, you collect it, we bring it to the lab to dry it, and then we extract it. So we have to get the chemistry out of the plant so that we can actually put it into a petri dish or some experiment. So usually we use various organic solvents. So we use alcohol is a very common one that we use, different types of alcohol. Acetone, like nail polish remover, is a really good solvent that we use for some of our extracts. And so we basically make kind of a smoothie, for lack of a better term. We grind it up. We grind up the plant material. We put it in with the solvent, and we put it in essentially a blender and blend it up and let it shake and stir kind of overnight to extract out as much as we can. And that gives us what we call a kind of a crude extract that hopefully has all of the chemistry inside. Now, sometimes we also still rely on this ecological knowledge for how to prepare the plants, because some cultures use, they make teas or they make poultices where they grind it up and, for topical application. So there are very specific ways to prepare the plant. They give us clues to how we can access the chemistry as well. If there is a traditional preparation, we'll use that parallel to this solvent smoothie approach. From there, traditionally it's all about isolating presumably the one active molecule. So we use various methods to separate out the chemistry, because a plant could have thousands of different molecules in it. And not all of them are doing the same thing, obviously. So we have to tease that chemistry apart and make it simpler and simpler and simpler until we've identified, hopefully, the one molecule that is an antibiotic or an anti-inflammatory or what have you. We do mostly antibiotics now. And so we do, it's usually multiple rounds of separating out the chemistry, simplifying the chemistry, and then at every stage retesting it to make sure that it's, finding which pieces are still active and that guide us for the next step. And that has been the way that most pharmaceuticals that come from plants have been discovered for the last 100 plus years. Everything from artemisinin, the anti-malarial compound, the discovery of which won the Nobel Prize 5-6 years ago, to cancer drugs like Taxol and molecules like that. They all came through this, what we call bioassay guided fractionation. So we fractionate and separate the molecules over and over again while testing them. And so it's been a really reliable way of accessing the chemistry and finding these bioactive molecules. But that process could take, once you have the plant, it could take anywhere from months to potentially years to find the one.
So when you talk about that process of, like, let's say you have created that plant smoothie, now you need to figure out which elements to separate. Is it like a standardized process? Is it like you have to come up with unique thinking, problem solving at each step?
That's a great question. And so there's two ways that we kind of approach it. One is, so if we're testing a lot of different plants, or if we don't have a good idea of whether the plant will be active, we first see, does it do anything to bacteria? So we have a library of pathogenic bacteria. We have Salmonella, E. coli, Staph, a variety of ones that we've gotten from the US CDC. We just try the plant extract that we've made. Before we do any separation, we just try it to see if it's active. If we have a feeling that it's active, now we can use more information about the microbiology, the structure, the function of the bacteria that we're trying to kill, if we have a specific one in mind, to help guide our isolation procedures. And I think that's really fascinating. That's using, I'd say, more modern techniques where we can profile certain targets, because a bacterial cell is very different from a human cell. And we can take advantage of some of those facts, that their cell wall is made differently than our cellular membranes. And a lot of the antibiotics that have been developed target some of these key differences between a bacterial cell and a human or mammalian cell, we'll say. The more that we know about these targets on the bacteria, the more we can try to rationalize what sort of molecules, what sort of chemicals we should be hunting for. That'll give us an idea of what is known that fits that target. But we're also interested in new molecules, new structures, new chemistry that might hit that target or different targets that we don't know about. It's kind of a two-edged sword. We can use these targets and use what we know about drugs that already attack them or fit them or inhibit them. But that also potentially is limiting, because we're also interested in new targets. So bacteria, they are very well adapted to overcoming the treatments that we already have on hand. And that's one of the big problems that we're having with bacteria, is that they become resistant. So these mechanisms, these targets that we've been moving towards for the last almost 100 years since penicillin first was discovered, the bacteria, they reproduce so quickly, they evolve very quickly, and they have become really good at finding ways around our current treatments, most of them. And so what we call microbial resistance to our antibiotics. And so that's one of the reasons why we're hunting for potentially new targets, new ways to disrupt their basic cellular processes that keep them alive. And so some of that, we can team up with microbiologists to give us insight into how the bacteria works, and that gives us new potential targets to go after. Sometimes we kind of go blindly in and see, is this plant, are these chemicals, these extracts working against the bacteria? And we'll kind of sort out the mechanism of the action downstream.
Could you give us an example of some specific molecule or plant that your lab worked with, and that process from start to finish?
Yeah, of course. I will begin by putting the asterisk that we are a pretty new lab. I've been here at Penn State for just under 3 years. So, our research is still ongoing. But one of the projects that we've been working at has been with our collaborators at Worcester Polytechnic, which is in Massachusetts. And we're looking at the plant Artemisia. It grows in temperate areas from China to Russia to Europe to the United States and Canada and beyond. And Artemisia has the fame of being the genus from which artemisinin was found. Artemisinin is currently our best anti-malarial drug, candidate. And so artemisinin was discovered in the 1970s by a Chinese natural product chemist. Yu Tu is her name, and she won the Nobel Prize for the discovery of artemisinin. But we're not looking at malaria. We are actually using the same plant and looking at other respiratory infections. So we're looking at tuberculosis and actually COVID, because we've all been through this, and new treatments are never a bad idea. So what we've found so far is that the artemisinin, that anti-malarial compound, actually is not really super effective against tuberculosis or COVID. And so our research is geared at finding what other molecules might be in the plant that have been overlooked that are actually responsible for this activity against these other diseases. It's been really interesting to, as famous as artemisinin is as a natural product, molecule, and drug, there's more chemistry to be uncovered.
Yeah, it seems like the entire field is ripe for possibilities of looking back into the past and, figuring out some new application of what was. So what has the history been like of ethnobotany, of Western scientists actually working with indigenous cultures and What significance does it have for your lab and your work in particular?
That is a fantastic question. Speaking broadly, I would say the long history of Western scientists working with indigenous cultures in understanding this traditional ecological knowledge, the history has been more, I think, of extraction rather than partnership. There are numerous cases where, unfortunate cases where Western scientists have gone to engage with communities, but they take their notes and then they go off and do the research and collect the plants. And the community and the knowledge is not, they're not active partners in the research. So the scientists have been seen to come down, find out what they want to know, get the plants they want, and then they disappear back to the West to their laboratories. And then discoveries are made, maybe patents are made, drugs could be discovered, and none of that ever comes back to the people who did a lot of the original work. I mean, they were the original scientists, right? Testing these plants, finding out the knowledge of these plants. And so, we are very cognizant of that history as we approach communities to work with. So we work with some communities here in the United States, some Native American communities. We've also been working on forging partnerships internationally with indigenous communities and universities in other countries. And so we work extremely hard on several fronts to make sure that the community is an active participant in the research. So some of that is addressing their questions. What do they want to know about the plants that they use? Maybe they have, we often encounter that they have questions about environmental pollution and toxicity and how that impacts the plants that they are collecting and still using to this day. And so we want to make sure that, the research is tailored to be of a benefit to them as well as us. And then it's also, again, we're a newish lab. We have not had any artemisinin kind of breakthroughs yet. But if we were to come across a molecule that was, a super antibiotic or manifested some sort of really positive benefit, we have the structures in place that they share in our success. From an academic standpoint, that could be co-authorship on scientific papers and conferences, but it could also be sharing of the discovery and any patents moving forward. In addition to just from an anthropological, from a societal anthropological standpoint, from the plant standpoint, there have been more and more restrictions put in place to prevent that sort of extraction from tropical countries, countries around the world back to Western countries. So export controls have been put in place, so that it also helps make sure that the benefits can stay with the country, with the communities, which is important. If we export plants from another country, we have to have permission from the community as well as the government to actually bring those plants back to the United States to do research. It makes it more complicated. There's definitely more hurdles to jump, but I think it's still important because we want to make sure that we are protecting everyone and making sure everyone has a voice in the research. It's been an evolving process, I think, but I think it's getting better. But we definitely still see a lot of distrust when I am a white male from a Western fairly first world nation, so to speak, and coming into other communities, there is distrust because there have been people and researchers in the past who have not behaved ethically. It's on us to make sure that those relationships are nurtured and continued.
Yeah. If I may ask, do you have any memorable conversations that you've had with some of your partners about where they've helped you discover something new that you couldn't have previously imagined?
Ooh. Some of my work, and this was actually as a graduate student, was with Alaska Native communities. We were focused mostly at that time on nutritional plants. And so there was definitely, on the one hand, we did encounter a lot of hesitation from the communities about interacting with white scientists from the lower 48, kind of, as outsider as you could be in a way from these communities. And so we definitely had to work hard to describe the work that we were doing. We came back many times to share our findings with the community. So we went back year after year to give them updates on what we did. So we gave talks and shared data and talked to them about what we had found. it was a long process, I think, to get them to open up to us and share their harvest sites and their knowledge. And even when we got close to publishing, they requested, don't use the GPS coordinates because we don't want people knowing where these resources are, because they were afraid of others coming in and over harvesting and consuming them. So on that side, it was a really interesting process of gaining their trust. And it taught me a lot as a grad. I was a grad student at the time. As a grad student as well, I worked on teaching workshops that bridged U.S. universities, foreign universities, and local communities. And so we held these workshops in various countries across the world. And there were two that stood out for me that I think really hammered home the importance of this knowledge. The first one was in Ecuador. It was one of the first workshops that I taught. And we had already established a relationship. We were chatting with the communities, and we were going to head out and do some field work, identify some plants and take a look. It's still, to think about to this day, it blew me away that these community members, these traditional healers and elders in the community, they could walk down the path. They'd walk down, and every couple of feet, they'd be like, yeah, we use that for stomach parasites, and this is good for wound healing, and this is good for fever. It was literally like walking down the forest path was walking down a pharmacy for them. And the ones that we collected and worked on testing in conjunction with them and their university partners, they completely worked. And that's not surprising necessarily, because I trust their knowledge, but it was second nature to them at that point. Like, they were so intimately involved with their environment and this knowledge that it wasn't even a question of what plant is this or what does it do. Like, it was an instinct. The second one was actually a time when I got sick on one of these workshops. We were actually in Kenya, and I came down with some sort of stomach bug. I don't know, but it laid me out. I was basically lying on the floor of my hotel room, unable to go anywhere. And my co-teacher, we reached out to the folks who were in the workshop with us, and they had two minds. Some people said, "Oh, just go to the pharmacy, get ciprofloxacin or something for digestive infections, something like that, and just take some Cipro, you'll be fine." But then one of the traditional healers that was part of the workshop said, "No, don't do that. I got exactly what you need." And he had a briefcase that he pulled out, this blend of plants that I don't know what it was, but he said, "Brew this up as a tea, drink it twice in a day, and you'll be good." And it was interesting that even some of the people from the universities were like, "Don't do that. Just go get the Cipro." And so I had to step back and say, I was like, well, we're here because we value this knowledge and we trust this knowledge to do discovery and base our research on it. So how can I not trust it to fix my own, to try to fix my own health? So I went and I made the tea, and it tasted horrible. It tasted like dirt and mud, but it's sure enough, the next day I was back up and raring to go.
Yeah, I can see how it's an important cause for you, given that you made yourself as a mini test subject there.
Yep.
So I guess while we're talking about this right now in the world, there are lots of indigenous cultures that they're facing quite a lot of challenges with biodiversity loss, with the encroachment of traditional territories, with loss of language. How have these issues been affecting, the experiences you've been having?
These communities, unfortunately, I think are often at the forefront of a lot of negative changes in our world, right? So they are often at the point where climate change is impacting them the most, habitat loss, biodiversity loss, like you said, potential disruption of the ecosystem, or even pollution from other extractive industries. I'm thinking logging, mining, oil, petroleum exploration. And so they're bearing sometimes the brunt of this, as well as disruption to their own culture. You mentioned, loss of language, and we've seen, and other studies have shown that there's a loss of transmission of this knowledge from one generation to the next. So the elders are not passing on, or the younger members of the community are maybe not always interested in learning from their elders, especially communities that exist mostly with oral tradition. Once it's lost, it's lost. And this is, it's heartbreaking from a cultural standpoint, but also thinking as a chemist, as a biologist, as a scientist, thinking about the resources that we might never get a chance to explore with biodiversity loss, habitat loss, climate change. On the one hand, sometimes it feels like a race, right? We're not only are we trying to find the next antibiotics, but we're kind of in a race to document and preserve as much as we can before we lose it.
Yeah, it's a good reminder that discoveries, technology, science, business, all these things don't happen in a vacuum. So could you talk about some of the major people and new methods that have progressed this field forward over the last few decades?
Sure. It never fails to amaze me how scientists did this kind of natural product work even 40, 50 years ago. It doesn't sound too long ago. I mean, it's longer than I've been alive, but it's not that long ago. But the leaps that we've made in technological advancements and instrumentation to help us in this search has really been incredible, right? So we are much better able to understand the connectivity of atoms and how molecules are put together. So deducing the structure of a new molecule that you get from a plant or a fungus that no one has ever seen before, it's not necessarily trivial, but it is orders of magnitude easier than it would have been in the 70s. Our capabilities of detecting molecules as well as understanding their structure has made this field open to more scientists, but also it has removed some of those barriers to progressing from the basic science into next steps, which has been important. You need to really know what the molecule is on a structural level before you can propose safety trials, toxicology studies, clinical trials, definitely. So we've also been better, our instrumentation has gotten better at getting more sensitive. So we're able to see that some of the smaller molecules, smaller abundance molecules in a plant or in a natural resource, sometimes are the ones that are most bioactive, but they, in years past, might have been missed because they were just not present in high enough quantities to actually produce a signal. And so the advancements in mass spectrometry and nuclear magnetic resonance, so two of our main staples in profiling chemicals as well as understanding the structure of chemicals, has really helped us in terms of our studies. As I mentioned earlier, plants have potentially thousands of small molecules in them at any given time. And so teasing out which ones are actually bioactive relied on this kind of iterative process of simplifying the chemistry and testing, simplifying it more and testing. And a lot of the pharmaceuticals that we see today have arrived by that process, but it is a long process. And so groups have been focused in how can we more rationally, how can we more efficiently search through plant chemistry, search through these natural products to find the chemistry that we are interested in. And so the advancements in instrumentation, as well as advancements in statistical modeling, artificial intelligence, machine learning approaches have been really key to helping drive this discovery forward. So the goal is that we can identify the chemicals of interest at an earlier stage, so we don't have to go through rounds and rounds and rounds. That means that we can speed up the process, but we can also be more selective in what we're studying.
If I could try and say that back to you, we've had better technologies to measure the actual structure of molecules. We've had more luck trying to measure, identify, work with smaller concentrations of molecules, and we've had better computing power to decide which molecules are worth testing.
Yeah, I think that's exactly it.
Final last question would be, are there any areas that you feel need more work?
We're looking at how can we better integrate different types of data. We can get the genome from a bacteria if we're looking for targets, or from a plant to understand what it possibly synthesizes, what molecules it makes. How can we tie that in with the chemical data that we're seeing of what it's actually making? And what is that telling us about the environment of the plant, the condition of the plant? So integrating these big data sets, there's already groups working on it as well. These molecules that are being made by the plants they're not for us, right? Plants don't make anti-cancer drugs because they think some human might eat the bark of a plant one day when it has cancer. So they have their own ecological needs that they're filling with these molecules. And there are a lot of cases where we don't know what those are. Plants don't get malaria. Why is a plant making a really potent anti-malarial drug? That I think is a really interesting question and that would help us from a medicinal or human health perspective. But I think it's also fascinating from an ecology perspective, understanding how plants fit with bacteria and fungi and insects and herbivores and, in this really complicated, complex environment.
I really appreciate you, taking the time to give a snapshot of what it's like, working with some of the concerns on your mind, some of the problems that excite you from a technological perspective.
It was a supreme delight to chat, and I loved your questions, and I had a fantastic time.